Hey guys, this is your learning assistant for Biology 1009. Today I have a short video for meiosis. So I'll be talking about simply what meiosis is, and I'll go through the steps of meiosis 1 and meiosis 2. So what is meiosis? Meiosis is a specialized form of cell division that produces reproductive cells such as plant and fungal spores, sperm and egg cells. So sexual reproduction occurs only in eukaryotes. Therefore, meiosis only happens in eukaryotes as well. During the formation of gametes, sperm and eggs, the number of chromosomes is reduced by half and returned to the full amount when the two gametes fuse during fertilization. There are two successive nuclear divisions occurring in meiosis. The two parts are there for meiosis, which is first one is meiosis 1, it's the reduction part, and then the second one is the meiosis 2, the division part. So meiosis produces 4 haploid cells, while mitosis produces 2 diploid cells. So here's another quick compare and contrast of between meiosis and mitosis. Meiosis makes 4 haploid, mitosis makes 2 diploid. The old name for meiosis, used to be reduction and division because, as you can tell, meiosis 1 is reduction and meiosis 2 is division. So meiosis 1 reduces the ploidy level from 2n to n, while meiosis 2 divides the remaining set of chromosome in a mitosis-like process, which is a cell division. And here is the well-illustrated diagram of meiosis. As you can tell, there are interphase G1 and interphase S steps before the prophase 1. These two steps here are not part of meiosis. These are just necessary for the chromosome replication to happen in order for the prophase 1 to start. So meiosis starts from prophase 1 and ends at telophase 2. So at prophase 1, centrosome movement, spindle formation, and nuclear envelope breakdown have happened, just like in mitosis. Chromosomes condense progressively throughout prophase 1, and during the early phase of prophase 1, this thing called crossing over happens. Might happen or might not happen, but most likely will happen. So crossing over may or may not happen at this point, but if it happens, during the crossing over, the chromatids break and may be reattached to a different homologous chromosomes. So, due to crossing over, new association of genes and alleles might happen because of crossover at prophase 1. And at metaphase 1, pairs of homologous chromosomes will be arranged at the metaphase plate. So, like this vertical line right here, not the vertical line here, but the horizontal line. So this will be the metaphase plate, and along with that line, pairs of homologous chromosomes will be arranged at that horizontal line, which is called metaphase plate. And at anaphase 1, the homologs will move toward the opposite poles guided by the spindle apparatus. So as you can see right here, anaphase 1, the chromosomes are going toward each end of the cell. So each poles, and they're going toward the opposite poles, guided by the spindle apparatus. And at telophase 1, something interesting happens. So when telophase 1 begins, each half of the cell has a complete haploid set of duplicated chromosomes. So each chromosome is composed of 2 sister chromatids. One or both chromatids include regions of non-sister chromatid DNA. And cytokinesis, which is a division of the cytoplasm, usually occurs simultaneously with telophase 1, forming 2 haploid daughter cells. And in animal cells, like the one here, a cleavage furrow might form. It should form. So this red line that I've just put right there, a cleavage furrow for animal cells will be formed, and for plant cells, it will be a cell plate forming, just like the red line here. So at telophase 1, cytokinesis will happen simultaneously, and then the cleaved furrow for animal cells will be created, or cell plate forms for plant cells. And at prophase 2, a spindle apparatus forms. And in late prophase 2, chromosomes, each still composed of two chromatids, move toward the metaphase 2 plate. So, well, here at metaphase 2, the chromosomes are aligned on the metaphase plate, but at late prophase 2, the chromosomes will slowly move to make that line at metaphase 2. For metaphase 2, I mean. So at prophase 2, chromosomes at late prophase 2, chromosomes will slowly start moving to make the metaphase 2 that line. And at metaphase 2, the chromosomes are positioned at the metaphase plate as in mitosis, and because of the crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. And at anaphase 2, breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate, and the chromatids move toward the opposite poles as individual chromosomes. So as you can see on the diagram here, the centromeres allow the chromatids to separate. So they are separated, and then each individual will be moving toward the poles, at the end, the opposite poles. And finally, at last, which is a telophase 2, nuclei form, the chromosomes begin decondensing, and cytokinesis happen as well. So the mitotic division of one parent cell produce 4 daughter cells, each with haploid set of chromosomes. The 4 daughter cells are genetically distinct from one another and from the parent cell. So that's about it. As you can see, after telophase, there are 4 daughter cells that are haploid. If hopefully my video helped, but if my video didn't help, here's the really nice website. Here's a link. This website has a video, more clear video, more professional video about the steps of meiosis. So I recommend you to check this website as well. And yeah, this is about it. Thank you.